여기부터 저길 가려.
Ladies and gentlemen, my name is Lim Chong-yun, the chairman of the board of Korea Biotechnology Industry Organization. We are commenced by 20, 30 years ago, government under Ministry of Industry, and industry put together an effort to a proliferate bio-industry and future of Korea technology. It's my great pleasure to welcome everyone 2021 Global IR Pitching at JPM on behalf of three co-organizers, KoreaBIO and BioCentury and Sidley Austin, and not to mention the Ministry of Industry, our main sponsor today. Today we'll try to be a little unique, but nowadays it's a little bit common. It's going to be a virtual session and opportunity to raise awareness of the important bio startups, acceleration, but more to an ecosystem, building an ecosystem under post-pandemic and very fast-growing Korea bio industry. Through this event, we hope to see more bio startups change the industry, great innovation. We believe a global IR pitching session like this has to go on under pandemic situation, under any circumstances, because this is probably the biggest protection we can have under certain situation like this. And then probably in Asia, we have fast accepting technologies and fast growing industry that can cope with the experience from the US or Europe and many investment bank and also organizations. Today we will have 8 pitching sessions prepared by venture companies in Korea, and also we will have a few more industrial and regulatory specialists to share the information and also promote our acceleration program that prepared by many people has put effort on this one. So we're looking forward to hearing more policies and more innovative approaches to the regulatory systems in Korea. And probably right now we have going under very rigorous changes in the regulation, and probably a lot of new and hopefully it's beneficial to us to have promote more innovative, more challenging and more pioneering technologies will be blossom in Korea. I have no doubt about this one. So even though we proceed with the virtual sessions, we'd like to thank all of the people who has put many effort into this one under very specific situation. And we'd like to thank all the sponsors, including the Ministry of Industry in Korea. I will now hand the session over to the session chair and the panel discussion, Mr. Jeffrey Kramer, Executive Editor of BioCentury. And I hope everyone enjoys this one and then try to become a fantastic discussion with the industry and investment and people who is working for regulatories. Thank you very much.
Look for cross-border collaborations in the year ahead. As the Korea biopharma industry matures, opportunities are increasing for cross-border collaboration and investment to accelerate drug development. We have a great panel here for you today. Given that we only have a short amount of time, though, I thought we would skip the introductions and I would introduce each panelist as we go along. I'm Jeff Cranmer. I'm an executive editor at BioCentury, and I'm sitting in the United States where we have a new president today. And so I thought that it would make sense to head to Washington for our first question to an alum of FDA, Becky Wood. From your perspective, Becky, on Sidley's Global Life Sciences Leadership Council and leader of its DC Healthcare and FDA group, as well as former chief counsel at FDA, what are your expectations for the Biden administration in terms of policies that might affect Korean biotechs, the Korean life sciences ecosystem, and cross-border deals?
Well, Jeff, it's such a pleasure to be here with all of you today. Thank you very much for the invitation. We, of course, are watching all of those issues very closely for our Korean clients and for all of our clients who are FDA regulated or interested in becoming FDA regulated. And it is, of course, very timely that we're having this conversation today. There are seismic political shifts underway in Washington, D.C. today. Of course, America inaugurated Joe Biden as a new president, bringing a Democratic administration to the executive branch of our government. And what that will mean is, of course, political appointments at the helm of all of the federal agencies that regulate the life sciences industry, including FDA, as well as a new Democratic Congress, which is going to have a lot of implications in terms of investigations and focus going forward in the next couple of years. And as you know, the conventional wisdom is oftentimes that Democratic administrations tend to be somewhat less friendly to industry and innovation than Republican administrations, and also more pro-regulatory. But I think the landscape is much more complex than that. So we're going to see some significant changes overall, I think particularly in the tone that we see from this new president and this new administration. But I think people would probably be surprised that there's also probably going to be a lot more continuity than you might expect at FDA itself. I think it's important to remember that FDA itself is a science-based organization. It regulates about 25% of the US economy, including, of course, biopharmas and medtech. And it is largely led by a really established and well-regarded set of career personnel. So all of the heads of the different device, biologics, and drug centers at FDA, our career personnel have been there a long time, and they are not changing. So there is also going to be a continuity story that we're watching very closely. You know, FDA operates very much like an independent agency. It is tasked with, imposing Congress's mission and making sure that the products that it approves are safe and effective. In the last year in particular, I think we have seen significant concern in many quarters that the president and HHS were perhaps interfering or trying to interfere with science-based decision-making at FDA. And so you're seeing in reaction to that, even today, as priority point number one for the new Biden administration, I think a real focus on a new tone. And they have talked very much about listening to science, ensuring that public health decisions are informed by public health officials, and promoting trust, transparency, common purpose, and accountability for the federal government. So I think you're going to see a change in tone. Frankly, in the day-to-day work of the career personnel at FDA, things may not change as much as folks think. And I want to focus on a couple of things that I think are pretty interesting. So of course, COVID is a number one priority, both at FDA and for the new administration. A continued rollout in what has really been an extraordinary, unprecedented effort to bring vaccines to market at lightning speed. So new vaccines, new therapies, new diagnostics to make sure that you detect, treat, and prevent the spread of COVID, and also focus, of course, on personal protective equipment. All of these things have an FDA regulatory component. We're continuing to see a lot of questions from clients about how to successfully navigate the emergency use authorization process at FDA. And obviously a focus as well, we think we're going to see from this administration on science, elimination of political influence, they say, enhanced transparency, they say, and continued regulatory flexibility, which has, of course, been a hallmark of FDA in recent years since the 21st Century Cures Act, which really put a focus on modernizing the way FDA regulates in order to speed the marketing of important new products. So that sounds in some ways quite similar to the Commissioner Gottlieb and Commissioner Hahn FDA, but I think a real change in tone from the top. There's also, of course, been a focus on COVID as a catalyst for moving really important policy initiatives and regulatory flexibility, and we're continuing to watch how that affects non-COVID therapeutics development. And so, just to name a couple of examples, in terms of the use, comfort around the use of real-world evidence, comfort around enhanced flexibility for clinical trial, and digital health. Those are all things where the agency has been somewhat reluctant to jump with both feet. And COVID has, I think, really encouraged novel thinking and practical thinking that will inure to the benefit of all innovators in the system. In terms of novel therapies, of course, the 21st Century Cures gave FDA these new authorities. FDA has already stood up a framework for being more nimble in how it is regulating, providing for accelerated approval of really important therapies for novel and rare diseases in particular. And, you know, it's interesting, FDA put out its scorecard for 2020, and it actually approved as many novel therapeutics last year as it has for the last several years. It's the second to the record in new therapy approvals. And the FDA also at CDER, the drug center, met 100% of its user fee deadlines, which is fairly extraordinary given the press of work with respect to COVID. Interesting thing about that is, though, we heard from a lot of clients that felt like they were getting less robust interactions on complicated questions with the agency. The agency was more apt to perhaps issue complete response letters where it had questions. So a little bit of two sides of that story, I think, as you think about innovation. Gene therapy, another issue we've been watching very carefully. FDA, of course, stood up a framework for modernizing that. We've had the first gene therapy approvals when I was at the agency a couple of years working with Scott Gottlieb and Peter Marks in developing that framework. And we're seeing a lot of focus on gene therapy and particularly challenges around safety. We've seen clinical trials stopped. We've seen challenges with scalability. So folks focused on that are obviously very concerned about continuing to have flexibility in those areas and also the support they need in that incredibly complex but incredibly curative and innovative industry as well. For drug pricing, that is obviously an issue that got a lot of attention from this administration, a lot of attacks on pricing, efforts to allow the importation, which is a new thing, of drugs from Canada, for example, and personal importation. And the Biden administration has said, you know, that drug pricing and bringing that down continues to be a major effort for them. We also are watching very carefully what may happen on the compliance and enforcement front, which when you're innovating and you're getting into a market can seem like it's an awfully long way away, but it's a very important area when you're doing diligence to figure out, you know, what are the potential problems and what you might be buying or starting. So that's been an issue where we think we're going to see as FDA opens up inspections out of the US to manufacturing facilities. We're going to see more emphasis on that. And of course, a bad inspection can derail an FDA regulatory approval. As you think about longer term risks to business, we're also looking in this administration for a resurgence of enforcement, particularly targeting alleged COVID-related fraud, warning letters for overly aggressive promotional practices. That has been, in particular, the latter, a hallmark of Democratic administrations. I think we're going to see more of a return to that as we look forward to what's next. And finally, I'll leave with congressional investigations, you know, and having an aligned Congress and administration, both of the Democratic Party, you're apt to see a lot more investigations, possibility of CEOs getting hailed in front of Congress for uncomfortable questions, and a real focus in particular on perceived fraud and abuse with respect to COVID countermeasures and COVID response. So fascinating time here in Washington and a lot that we're continuing to watch.
Excellent. Thank you for that very detailed response. And the points on cell and gene therapy were quite interesting, as we have Kun-Woo Lee on the panel, as well as BG Ree, who are both working at companies that are well-versed in one or the other of those areas. And you did mention, you know, it's changing, but it's not changing. I just wanted to point out that on that note, the Biden administration announced today that Janet Woodcock, who is the longtime head of CDER at FDA, will be the acting FDA commissioner. So in a way, consistency. Good to see at FDA. Let's dig right into cross-border deals. I'd like to turn to BG in Korea, the CEO of SCM Life Sciences, no stranger to cross-border deals. BG, what do you see as the biggest opportunity for Korean biotechs as they look outward to form cross-border deals in the year ahead?
Well, year 2021 and the year ahead, we have two big issues. As Becky mentioned, there's one is COVID-19, and the other one is the Biden care. So for COVID-19 case, Korea did an excellent job. For the diagnostic kit, we have more than 70 companies manufacture the diagnostic kit and exported 50 more million kits to 110 countries. Okay. Also, with combining with Korea's good IT system, we did a good job for the tracing and the prevention for COVID-19. Korea also shows we have good health hospital system, and we can conduct excellent clinical trials. And for Biden care, maybe biosimilar will be one of the part, and Korea is globally #1 in biosimilar industry. In Samsung and Cellatoz, they are expanding their CMO capacity from currently around 550,000 liters to 930,000 liters. That's mostly the biggest capacity in the world. So we can utilize this kind of thing. So in the coming years, KoreaBIO industry can be much more active for cross-border the partnership. Also in the novel therapy area, Korea is very much focusing on regenerative medicine, including cell therapy and gene therapy. Also, we are working for microbiome area and the exosome area. So that, Korea can go globally compared to other industry. Korea has always some kind of discount for cross-border partnership because we have a small market and the bio industry was not great industry for Korean peninsula. So we are aiming, we want to become maybe hub for the Asian territory. The Asian market will be growing dramatically in the coming years and try to become a hub of the Asian territory for later. So Korea has a lot of good opportunity for this coming years, and we hope we can catch that.
Chris, do you want to jump in there? I know that you've been watching investments in U.S. companies. You can speak to investments in Korean biotechs as you are partner of Novatio Ventures and managing partner at the Korea Seoul Life Science Fund. So curious to hear your thoughts there.
So given the backdrop of what we all had experienced last year, it obviously was the worst year. But in terms of biotech equity market, it turned out to be the best year for both Korean domestic market as well as U.S. venture community. And what I see is that given the fact that in South Korea, the venture investment in biotech has actually over the last 5 years has increased by fivefold. So that's a significant increase in VC funding in Korean biotech, domestic biotech. But what has happened is, as far as I know, to my knowledge, there is a very small, almost non-existing amount of U.S. VC invested in Korean biotech companies over the last few years. And our Korean fund actually has been kind of unique in a way that two of our portfolio companies had successfully attracted VC investments from very marquee, very well-known US VCs. So that was unique, but ever since it sort of dwindled, the US VC investment in Korea has become diminished significantly. And I see that as a potential opportunity where it can be sort of brought back because as you are probably very well aware, US VCs, especially early stage focused VCs, tend to be very hands-on. So there may be some conflict, cultural conflict, if US VCs were invested in a Korean company, but what US VCs in exchange bring is a very significant level of experience and in helping the company realize its R&D, long-term R&D strategy. So I think there's a significant benefit of having partnered with US VCs. And also, US VCs, as you know, really tend to gravitate toward high risk taking, and with a lot of money that's available from LPs nowadays, their appetite for risk taking has significantly improved, and I think if US VCs were to invest in Korean biotech companies, I think that provides a very positive signal as to the validation for the company's underlying technology and management. And I would also say, perhaps lastly, the Korean biotech companies, so they are very self-sufficient in a way that Korean capital market has been such lucrative and active over the last few years. Companies were able to raise money and go public, and as a result, management, has done well, in terms of, exit and all that. But again, what you should not really lose the focus of is the fact that biotech R&D strategy is global. So in order to realize your global vision, which I know that a lot of Korean biotech companies have, is one of the way to realize that is partner with US VCs.
Thank you for that, Chris. Kun Woo, I saw you nodding along there. Your company, GenEdit, is based in the Bay Area. I believe you're just across the bay from me right now. And you attracted a Korean investor, SK Holdings, as the lead for your seed round. I believe you have a couple of other Korean investors. What resonated with you that Chris said there? Nope, I believe you're on mute.
First of all, I want to thank the organizers inviting me and giving me a chance to speak with this wonderful panelists. And I have noted a lot. I mean, things that I have heard so far from the Becky and VG and Chris, and especially the parts that Chris has mentioned. There are different values that U.S. investors bring compared to Korean investors, and all different opportunities. To many of the entrepreneurs who are running the company, I want them to deeply consider that. And GenEdit, in our case, we were very fortunate to have both types of the investor investing in the company and adding value to GenEdit. Maybe sharing a little bit of our story, how the company was started, and company was started with the technology discovered at UC Berkeley, and the very initial seed funding came from Sequoia Capital and the Bow Capital. Bow is backed by University of California system. And with those money, we have started the company, and additional investors joined our round, which was Data Collective Bio and SK Holdings. And that was the first time we got the Korean investors' money into our company. And we haven't publicly disclosed, but in our Series A round, we have brought many fantastic investors, help of the investors from US side and help of the investors from Korea side. We have built really great syndicate of the investors there. And so in this environment, there were lots of things that we have seen, and there were parts that was significantly adding value. As Chris has mentioned, US investor has takes risk in the technology that is in early stage, and there's help the company build the technology further. And there's at the same time Korean investors are very supportive of the leadership team, and then talk with the direction, and then support the companies making continuous progress. So there are multiple parts that we are seeing, and maybe just adding a little bit more of the story to it, so that as many of the entrepreneurs are in this Zoom setting, because in this environment, bringing many different investors, very important part for us, building a relationship and really knowing, and also knowing each other and telling the story that we have and sharing the data that we have like collected. So that was like kind of very important part for us. And yeah, regarding the difference of the US investor and the Korean investor, yeah, like when I have chance, I will talk more about it.
Excellent. Well, I'm curious, perhaps this is one for Chris, but I know BG and Kun Woo have had experience here, and I'm sure Becky can probably just answer just about any question given her, the depth of her experience. But if you are a Korean biotech and you want to attract US investors, what do you need to do? Is it advantageous to establish a US subsidiary? What advice can you give to Korean biotech that's looking to attract U.S. investors, get a start in the U.S. market?
Okay, so I'll go. So I guess two things go hand in hand. Obviously, it's underlying science and the management team. And as mentioned earlier, U.S. VCs tend to take a lot more scientific risk. So if you have truly a breakthrough innovation that you can showcase, obviously that's going to help a lot. And management and team. So disadvantage of being in Korea and wanting to raise money from the US VCs is the fact that your exposure, your visibility in US is inevitably is low. And that's something that you have to overcome. So, some biotech companies come here to open up an office and start, one of the goals that they want to accomplish is relationship building. And I think that's important, but I think it's even more important that if you as a Korean biotech company want to come to US and open up an office, I think there has to be more practical and operational and substantial rationale as to why you are doing it. And meaning that you are doing it in order to ultimately help accomplish your strategic R&D long-term goal. And by definition, once you accomplish that, I think you will become more robust Korean biotech company in US, and that will in return help attract US investments.
Excellent. Well, we just have a few minutes left, unfortunately, as this is an excellent and insightful conversation. I'd like to head back to Korea to ask BG our final question. BG, STM has expanded into the US and Europe. What are the advantages of these deals for your company as a lesson to other companies who might want to follow that same path?
Yeah. We acquired the company in US that's for the mRNA-loaded dendritic cell cancer vaccine company. Also another company in Italy that's for allogenic CAR-T CD9 company. The reason we acquired the company is still Korean biotech industry has some gap for the global standard in cGMP manufacturing and clinical trials at global level. So the reason we acquired the company is we can acquire all kinds of know-how for the manufacturing and the clinical trials by acquiring the companies. So that's good asset for the Korean biotech. We are going to bring that kind of know-how and the information to Korean biotech companies to become the global level standard. So that's one of the main reasons, and we got this kind of thing very easily by acquiring the companies.
Excellent. Well, I think that's all we have time for. Actually, I want to throw in one last question. This was one question that came from one of the companies who's joined us, and I'd like to go back to Becky. One question was very specific about IP. Do you have any advice for Korean companies looking to patent their IP in the US?
Yeah, it's a great question, and it can be a neglected area. And I would heartily encourage people as part of their initial team to, in addition to getting your regulatory consultant, also make sure you have an IP patent lawyer as part of your team. And I think another thing that's very important to understand in the US system is that in addition to the patent law protections, there are also certain protections that are offered as a matter of FDA law. Where you have, for example, an orphan or certain innovations. And those requirements are exquisitely complex, but essentially they can stop, as you know, another company from having their application reviewed by FDA for a period of time. And sometimes those things are neglected until later in the day, and that may reduce the amount of opportunities you have for planning. So I would definitely think about those things proactively from the beginning and figure out how you fashion your patent and exclusivity portfolio along with the other important decisions about what track you're gonna do at FDA.
Excellent. Well, thank you so much. I'd like to keep us on time here. I'd love to hear more from all of our panelists. But now I'm going to say thank you for all your insights and to everyone who has tuned in around the world. Now I'd like to hand the microphone back over to BG, who will lead the company introductions. Thank you.
Thank you, Jeff. Well, I hope you all enjoyed this panel discussion. Now let's move into the global IR pitching session today. I'm BG. I am the moderator of this IR pitching session today. This IR pitching is to open investment opportunities for both Korean and global biotech startups. Today, I have 8 promising Korean biotech startups that aiming the attracting global investment and expansion. Each company will have a 10-minute presentation and 5 minutes of Q&A time. Please feel free to ask questions during the presentation by the chatting window. Let's meet the first company together, Abtis. So please welcome Justin Oh, CBO from the Abtis.
Thank you, Dr. Lee, and good to meet you online. And it's a great pleasure to make a presentation for Abtis today. We are located in Korea, and the main office is located in Suwon, nearby Seoul City. And we have a Seoul office too. And we were created in 2016. We are focusing on the ADC new therapeutics development, and it was created in about 4.5 years ago. Our core technology is ADC platform technology with stable and cleavable linkers and applicable native to commercial and novel antibodies. So there's no mutation, no engineering necessary to antibodies. And also we have the site-specific conjugation. So it's applicable to therapeutic ADCs and also companion diagnostics and long-acting biopharmaceuticals in the future. The founder and current CEO, Dr. Chung, is also a professor at Sungkyunkwan University in the pharmacy department. And he has experience at Harvard and KRIP and Scripps in the background. And also Dr. Choi is a CSO. He is a medical doctor and also PhD, graduate from Yonsei University Medical School, and has experience in Merck, GSK, and LG. So we have a strong management team, as you can see. And also we have strong SAB members, Dr. Joel Snyder, and also famous, well-known scientists and professors and medical doctors. We are founded in 2016 at Seoul BioHub, and we got the funding in 2018, Series A, and Series B actually recently closed this year, early this year. And during that time, we have been completing our ADC linker technology, and we believe it's the best technology, ADC linker technology in the world. We have a strong investor basis, leading venture capitals in Korea, Stonebridge, K2, and also asset management companies like Timefolio. We are striving to achieve our goal through our open innovation policy. We have already have several partners for the co-development of our ADC pipeline together, and also looking for licensing in and licensing out partners throughout the world. Our key technology is called AppClick, and it's the world's best site-specific ADC linker, we believe. And it has 2 simple, clean steps compared to the other ADC platform technologies. And linker conjugation and drug conjugation in 2 steps, and we have a complete ADC product. I'll explain this technology more in a later part. And compared to the native antibodies, our Abtis ADC has FcRn binding maintained and biological half-life maintained and similar circulation and excretion. We have also recently published peer-reviewed paper. Many of you well know about the ADC, but I'll go briefly explain about the ADC. And it has 3 components: antibody, payloads, and linkers. And the market has been really growing fast in the past couple of years. Since 2019, 5 ADC products have been approved by the FDA. So we have a total of 10 products available these days. And as you can see, the market has been growing very fast, especially in the approval of NR2 by Daiichi Sankyo recently is expected to be the blockbuster products in a short time. And also Daiichi Sankyo also had a multi-billion-dollar deals with AstraZeneca, totaling more than $6 billion each. And also global CDMO companies like WuXi and Samsung and Lonza has a strong ADC business also with ADC biotech companies. As you can see, Daiichi Sankyo has the NR2, and they have ADC pipelines, and the stock prices of Daiichi Sankyo is growing quite rapidly in the past couple of years. And ADC has been in a very hot area, especially in the second half of last year, like Gilead acquiring Immunomedics with a 108% premium. It's a total of $21 billion US. And Merck made an equity investment and also has a licensing deal. And also Merck acquired WellsCare. And also Boehringer Ingelheim acquired IMB Therapeutics. So ADC is a very active in investment wise also, and capital market wise. So there are big interest in ADC companies. So to explain our technology and our company, we call ourselves as a 3rd generation ADC technology company. And the first generation ADC company is Kesala, which was produced by Genentech, Roche, also Acetris by Seattle Genetics. They are in the market, and they are being sold very well actually these days. Kesala and Acetris have problems, have issues because they have a random conjugation. As you can see on the left side of the pictures, you can see the payload and the linkers are conjugated to the antibodies in a random ways. So it is impossible to control the drug antibody ratios too. So it is non-specific conjugation, and so it has problems of inconsistency of a drug antibody ratio too. So it has problem about production wise, obviously because of the random conjugation of that characteristic of 1st generation ADC. To solve the problems of 1st generation ADC, 2nd generation ADC companies recently have been developing site-specific conjugation. So you can put the linker and the payloads on the specific side of the antibodies. So the advantage of this technology is that you can site-specifically conjugate your payload and the linkers on the specific part of the antibodies. So you can control the drug antibody ratio, and you have advantages about that. But the problem with this technology, we believe, is that you have to mutate, you have to engineer your antibodies to achieve that site-specific conjugation. So there is also we believe there's might be some possibility of immunogenicity due to changes on antibodies. As you can see in this picture of rabbit IgG1, there are many license and many systems are there on these antibodies. So it is very difficult for the site-specific conjugation. Our third generation ADC has advantages over the first and second generation ADCs. We can control drug antibody ratio, new mutation or modification on antibodies are necessary, and site-specific conjugation to lysine in certain part of lysine on antibodies in Fc domain. So it's a Fc binding peptide linker is conjugated, and also it's a traces linker. So our advantages are like drug conjugation to unmodified antibodies. This is applicable to the therapeutics antibodies with no mutation. And this means also very cost-effective ADC production possible. And site-specific conjugation is a covalent binding to Fc domain and high yield of conjugation and simple process of purification. And this means CMC advantages of our ADC development. And the payloads diversity are also available. We can control the DAR, drug antibody ratio. We have 2 and also 4, and more numbers are also possible too. And also, the linker itself is very stable in serum, and it will be cleaved by specific protease within when the ADC gets into the cancer cells. As I mentioned earlier, we have a simple 2 steps for the ADC conjugation and ADC development. Site-specific linker conjugation to antibodies through specific part of lysine, and it's a non-covalent binding interaction by molecular recognition, and then the covalent bonded Fc binding peptide to lysine on the antibody with a click functional group. And the second step, the last step, is addition of payloads, drugs to the linkers by toxin conjugation by click reaction. It's a simple 2 steps, and no changes, no mutations on the antibodies are necessary, which is our biggest differentiation, strong points about our ADC platform technology. We have a strong IP strategies. We use US patent farms, and also we use Korean patent farms. These are our 3 major pipelines. We strive to be best in class in the therapeutics development. Our first pipeline is targeting gastric cancer, and we are planning to get into the preclinical earlier this year, and also getting the IND filing later next year. This is our whole pipeline at this time. As I mentioned earlier, we are striving to have open innovations through our partners. As you can see, we have antibody companies like iProgen, SG Medical, and Abtis. They provide their antibodies, and we provide our ADC linker technology. Together, we can create new ADC pipelines together. And also, we have our own pipelines. The first one is using the Herceptin HER2, targeting antibodies and new payloads. This is the animal study data, and we are trying to prove that our linker technology is very feasible and viable. And it shows that it has the proof that our anti-cancer effects of our ADC using Herceptin DM1 payload with a drug antibody ratio of 4 is working. So in the future, our technology has also applications because of using our platform technology. We are not only developing a new ADC, we also is planning to have an in vivo radioactive imaging platform with on the radioisotope payload, and a long-acting biopharmaceutical platform is also possible using peptides or protein drugs. And lab on a chip is also possible using our technology. We had a meeting with one of the 2 biggest semiconductor companies in Korea, and also they are interested in lab on a chip using our platform technology. This is our corporate milestone, and we recently closed our Series B with a strong investor support, and we are getting to the preclinical trial this year and IND filing next year, and for the phase 1 clinical trial in 2023. And also we are planning to go for the IPO using the technology exception track in Korea. And the last word is we have a strong ADC platform technology, and then we have a strong management team, and also our pipeline. We have with our partners, so we have a new pipelines also in hand, and thus we have a strong financials with the recent closing of Series D and our strong investors. Thank you very much for listening. Any questions?
Well, thank you for a nice presentation, Justin. Now, the audience is now open for question or comment. Maybe you can type your questions on the chatting room or raise your hand. Well, if not, we have a simple question, because you were in the capital market before you joining this company. So you completed the Series B funding very successfully this time. And would you give the market situation currently for KoreaBIO industry?
It is a very easy question, and also at the same time it's a very difficult question. It depends on case by case. I mean, VCs, I mean, I used to be a venture capitalist, so you asked that question, I imagine. And the VCs are looking for the, obviously, the really distinguishable technologies, technologies that is better than compared to the other technologies, have a strong implications in the market and or in the development side of the technology. So our ADC linker technology has that advantages. We believe that venture capitalists believe that, and also strong management is also very important. That the team has been together from the foundation of the company, and also we have Dr. Choi, who is a new CSO. Also, he has been. He will play a very important role for our development side too.
Okay, thank you very much.
Thank you.
So the next presenting company will be Cellatoz Therapeutics, and Dr. Jun Lee, Senior Executive VP, will present.
Well, welcome everyone. I'm Jun Lee from Cellatoz Therapeutics. As you can tell from our name, Cell A to Z, we're a cell therapy company in preclinical phase based in Seoul, Korea. Before I dive in, I want to share with you this slide. It reads different process, different results, and actually different product. In cell and gene therapy drug development, unlike the traditional ones where the CMC and process development happens during preclinical stage or later, we believe that in cell and gene therapy, process development must happen in the discovery phase to ensure that the cells that are used during the development process and also in the commercial phase remain intact and the same. So with that in mind, we have completed a cGMP facility to support all our developments and the early stage of our commercial launch. One more thing I want to highlight here is the RMAT designation, where if you're in an intractable disease area with the RMAT designation and Sakigake designation in Japan, the path to the market is much shorter and cheaper. Cell therapy 1.0, this is the first generation cell therapy development. In that time, the more focus was on the stem cells themselves, where they're originated from, how they're separated. So when you look at MOA, it's mostly explained as immunomodulation. And most of them were autologous and not live formulation, so COG was quite high. And whenever a donor was involved, there was a variation in efficacy due to lot-to-lot variation. But in cell therapy 2.0, the companies are more focused on the functional cells that are differentiated from the stem cells. So you get to see more clearer MOA and actual regeneration happening, and also changes in the microenvironments. And the products are mostly allogeneic and frozen formulation, so off-the-shelf products are possible now. Now, how does cell therapy 2.0 and Cellatoz are different? Well, for one, we have a confirmed CMC confirmed CMC that ensures with good quality control that the efficacies will remain consistent. And more importantly, we're a cell therapy company that actually owns a proprietary cells that have composition of matter patents. This is summary slides of the two programs that I will introduce today. One is musculoskeletal stem cell. It's an IP-protected cell that can differentiate into different musculoskeletal systems such as cartilage, bone, muscles, and tendons, etc. And it's derived from human embryonic stem cells and even iPS. An initial target indication for this cell is osteoarthritis of knee. The second one is neuronal regeneration promoting cell, called NRPC in short. It's derived from tonsil MSC, and it helps the regeneration of nerves. So our initial target indication here is Charcot-Marie-Tooth disease. And if successful, we think we can expand into other peripheral neuropathies and even into spinal cord injury. I'll introduce MSSC first to you. MSSC is protected by a series IP, including a composition of matter patent. And it's derived from human embryonic stem cell, and we differentiate them into MSSC. In 2017, we filed a couple of patents, which are now approved in US, Europe, Korea, and Japan in 2020. And in 2018, Longacre's group at Stanford Medical School has identified the existence of naturally existing human skeletal stem cell from the bone marrow. These cells are similar to ours, behave similarly, but we notice subtle differences in our markers, so our cells are different. That's why we received an approval of our IP. And unlike this naturally existing human skeletal stem cell, Cellatoz cells can be mass-produced for clinical and commercial use. I want to share with you some of our test results. This is a mouse forelimb model. If you see the photos there, we've actually severed a portion of the bone so that no natural healing can interfere. And as you can see, the empty photos, there are no bone growth. However, with injection of MSSC, by week 5 and week 7, you can see the formation of the bones. And we believe the bones are of human origin. The photo on the right actually is more clearer because the green color stains are the cartilages that are formed in the knee joint, in the joints of the mouse, and those are of human origin, which means our MSSC that were injected actually formed and regenerated into the cartilage of the rat. This is a preliminary data, and the focus here is that we injected direct injection of our frozen formulation of MSSC into the knee joints. And the graph that you see, whether our MSSC was washed with PBS or not washed, you probably see the similar results there. So we still have a lot more work to do, but our target indication is osteoarthritis of knee, more specifically grades 2 and 3, using direct injection and not surgery. Because we believe the environments in the knee joints are favorable for our MSSCs to differentiate into cartilages only and not into some other bones or anything else. Now I want to switch gear to NRPC. Our target indication is Charcot-Marie-Tooth disease. So what is Charcot-Marie-Tooth disease? It's a rare genetic disease that are pretty well known in Korea because 2 of the richest families are suffering from this disease. Overexpression of PMP22 genes would cause the person to gradually lose motor ability and sensory impairment. And as you can see in the photos, the disease starts from your feet and hands and gradually works up the body. And it's very debilitating. And it's a rare disease, but 1 in every 2,500 people are afflicted by it, and about 1.5 million patients worldwide. And it's obviously you can get orphan drug designation in Korea and US. And the CMT1A is the most prevalent type of CMT. And for cell and gene therapies, fast development is possible using our RMAT designation in US and Sakigake designation in Japan. Just to give you more background to the disease, what is Schwann cell? Schwann cell is a cell that actually covers the exon like the sheath. And whenever a defect occurs in Schwann cells, demyelination happens, and muscle atrophy results, and then sensory impairment also follows. So in developing a therapy for CMT, we believe that decreasing Schwann cell defects and also decreasing muscle atrophy are the most important aspect in our mind. As I explained before, NRPC, neuronal regeneration promoting cells, help regeneration of nerves. And these are derived from tonsil MSC. And our goal here is to help use NRPC to help regeneration or remyelination of the peripheral nerves, and so that the motor abilities can be restored, and even sensory impairment can be reversed or slowed down. Let me show you our mouse test result. The Trembler J mice are made through point mutation, so the resulting mouse has actually a severe form of CMT1A. The video on the left is the normal mouse. It behaves just like any mouse. One in the middle, it's a Trembler J mouse, and you can see that it doesn't move at all. The mouse on the right is the same Trembler J mouse with one single dose of NRPC treated. It's about 12 weeks old now, and you can see that it behaves similar to the normal mouse. Now, this is a rotarod test result. You see, look at the graph in the middle. The white bar is a Trembler J mouse without NRPC, and you can tell that it just drops off the rod. But the bar in the black is the Trembler J mouse with our NRPC, and by week 10 or 12, it hangs on over a minute. So we can tell that the strength in the muscles have increased or restored. So what happened? In short, we think remyelination of sciatic nerves were observed, resulting in strengthening of muscles and increased mobility. Picture A in red is the exons, and the image B is myelin. And so image D is, you see red enveloping green, that's the normal mouse where myelin is intact. But image I, it's the Trembler J mouse, and you can see that this is completely dismyelinated. But image N, when we inject NRPC into the TRJ mouse, you see remyelination happening. You can see the green color enveloping the red, which are exons. We think this is one of the MOAs. As you know, that there are nothing approved for CMT, and if you see the drug development status list, that we're the only cell therapy company targeting a CMT. And we already have formed good relationships with the KOLs and the Patient Advocacy Network. And Susan from CMT Research Foundation has been a great supporter, and Dr. Choi from Samsung Hospital, who treats 90% of Korean patients, will collaborate with us in our clinical trials. Other specialists are also in contact with us. So our schedule for these two in our pipeline. For NRPC, we expect an IND to be filed before the end of the year, and we expect to enter into phase 1/2A in 2022. And MSSC, even though it reads IND filing in 2022, we are accelerating the work, and we expect to file an IND before the end of the year. We're a company established 3.5 years ago. We have about 45 people actually working mostly in R&D and GMP. And our CEO, Dr. Lim, he's an expert in cell and immunotherapy. He was instrumental in getting a first cell therapy drug approved in 2007, which are still popular in the market. And he was also an author of a second generation cell and gene-based therapies by Elsevier last year. That's it. That completes my presentation. Any questions I can entertain?
Well, thank you for a nice presentation. Any question or comment? If not, there's a simple question about the sarcomania tooth disease.
Sure.
There are many protein drugs or gene therapy drug is also developing.
Mm-hmm.
And how do you see the differentiate between your cell therapy and those drugs?
We understand that there are some clinical trials going on, but we think getting the right genes to be edited precisely is not an easy feat. So we feel that the patient must experience improvement in muscular strength and motor abilities, and I think our way of approaching restoring the peripheral nerves is a sort of a easier or a better way at this time. And I'm pretty sure later on once the gene editing becomes more precise, we can see those drugs in the market.
Okay, good luck.
Yeah, thank you.
Okay, so the next company will be GBIOLOGIcs, and Mr. Hyo-Sil Lee, Vice President of company, will present. Okay.
Thanks for providing this opportunity to present our company and also our product. I'm usually from GBIOLOGICS. We are developing this stable galectin-9, shortly SGA9, for KRAS mutant cancer. I'm going to shortly go over this, briefly introduce this product, S-galectin-9, and the efficacy of this galectin-9 toward the KRAS mutant cancer. Okay, galectin is lectin-binding proteins, and it involves in immune homeostasis. There's a lot of galectins in human body, and mainly there's 3 subgroups, and galectin-9 belongs to tandem lipid subfamily. Tandem lipid families, as you can see in the file, there's 2 different lectin-binding active sites linked together by amino sequence, and we call it linker. And as I mentioned, that we are very much among all these galectin families. We are interested in this galectin-9. As you can see, this galectin-9, natural galectin-9, as I mentioned, is linked by this linker, and this linker, this amino sequence, is very susceptible to protease that is easily broken, that these 2 active lectin binding sites separate, then you lose activity. So this linker part was, by engineering, we modified this protein by removing this linker part, and these 2 active sites put together, and we call it S-galectin-9, stable galectin-9. And it is about 284 amino acid sequence. And this material is patent registered, and we are in very early stage of this product development. Okay, from the articles you can search through that galectin-9 involves, there's many various activities galectin-9 involved, and you can see there's a lot of potential activities and potential indications. Among these indications, we are currently focusing on KRAS mutant cancer. And so I'm going to introduce about the galectin-9 to KRAS mutant cancer. What kind of activities that mechanisms of this galectin-9 has to for this KRAS mutant anti-cancer efficacy? We mainly have these 3 activities. First one is cytotoxicity. Also, you can find from the various articles that this galectin-9 has cancer cell apoptosis activities. The second one is cancer cell suppressing cancer cell metastasis. Also, you can find articles about this. In addition to these 2 general anti-cancer activities, we have this KRAS mutant cancer cell apoptosis activities. We are focusing on these activities for this KRAS mutant anti-cancer efficacies. In normal cell, when you administer exogenous galectin-9, it internalized by endocytosis, and it is involved in regulation of lysosome, that lysosome smoothly function its own function to degenerate material in body and use as energy. However, in KRAS mutant cancer, when you administer this galectin-9, it internalizes and then it accumulates in lysosome. Because it keeps accumulating in lysosome, it swells, vacuolizes, and then causes cell death. So we are focusing on this function of this galectin-9 for this KRAS mutant anti-cancer to develop as KRAS mutant anti-cancer drug. Okay, this is just to show this the study result published by Netherlands scientists. They done this using galectin-9 to see the KRAS mutant colon cancer activity. You can clearly see from this first article that this galectin-9 cause vacuolization and then reduce cell viability. And they also showed that this galectin-9 activity was by this lectin binding site, because when you add lectin to this galectin-9, the anti-cancer activity was suppressed. So also you can see it from this from these studies. They also compared this natural galectin-9 with the short galectin-9, the linker removed galectin-9. They showed that the linker removed short version of galectin-9 has more higher activity, anti-cancer activity. Also, from using these normal colon epithelium cells, shows that it doesn't have any effect on normal cell, only effect on KRAS mutant colon cell or non-polarized cell. This is also this Netherlands scientist article to just show you that efficacy of this galectin-9. When this KRAS mutant colon cancer cell was SC injected to a nude mouse model to have this ectopic xenograft mouse model, and then they inject this galectin-9, and it shows that the tumor volume was reduced. And then also the mouse survival was increased. And then there's no body change, which means that it's non-toxic. So from this study, it shows that KRAS mutant colon cancer, galectin-9 has efficacy, potential efficacy to KRAS mutant colon cancer. For this, after looking at all these studies, we are currently focusing on the KRAS mutant cancers, especially pancreatic cancer, because KRAS mutant pancreatic cancer, because pancreatic cancer patients, there's more than 90% of these cancer patients was caused by KRAS mutant in cancer cell. So we are mostly focusing on this pancreatic cancer, and this studies were done in Korea. And you can see that when this MIA-kappa-B, which is the KRAS mutant pancreatic cancer from this KRAS mutant cancer patient, then we injected to nude mouse to develop this xenograft mouse model, and we injected either our galectin-9 in low dose 0.5 milligram per kg or 5 milligram per kg, and then also there's a gemcitabine we used as a comparable drugs. Gemcitabine is currently the standard drug for pancreatic cancer patients. So we use gemcitabine 50 milligram per kg. And then another group, we also have the combination therapy, gemcitabine plus our product, galectin-9 5 milligram per kg. And the result shows that the size of tumor was reduced, mostly therapeutically, greatly reduced by as galectin-9 5 milligram per kg. And we can clearly see, even though the error bar is high, you can clearly see that with 5 milligram per kg, we reached this therapeutic index. And gemcitabine alone doesn't have much effect. But however, when you inject gemcitabine to escaline combination therapy, there's reduction was increased. Tumor cell reduction was increased. Also, in survivor, the escaline 5 milligram per kg or combination of gemcitabine with escarline, the survival was the highest. And then the monotherapy didn't much work. Gemcitabine monotherapy didn't work much. So from these studies, these preliminary studies, we can come to conclusion that our S-galectin-9 has a potential for KRAS mutant pancreatic cancer. As I mentioned before, we are in very early stage. The problem with this protein was that the protein was unstable. There's a low expression in our engineering. So we work hard to improve this production, and we are currently have a successful result. And we are in this the production development, and we are going to scale up to 200-liter scale up, and use this to for the non-clinical and clinical studies. And as I mentioned, we are currently in vitro and in vivo studies with the university partners or CROs. Also, we are currently made a contract with the company for the partnership. And up to previous studies, we are more focused on ectopic xenograft mouse model, which means that we injected cancer cell to SC. But from now on, we are more focusing on orthotopic mouse model, much more close to this clinical the cancer model. So we are going to study in vivo, in vitro studies. And as I mentioned, we confirmed efficacy using this the pancreatic cancer cell and colon cancer cell. Also, we filed use as a patent. Our goal is, as we plan to submit IND to US FDA in early 2022. Okay, up to now is about the production. And briefly, I'm going to go over the company, introduce our company. Our CEO is David Song. He has various experience in development of biopharmaceuticals and more than 30 years development and involvement in development of a lot of various proteins, drugs, as well as vaccine. Also, our team members all has vast experience, more than 20 years experience in development of pharmaceutical product. Our company was established in 2018. However, the product was actually, escarline was acquired in early 2016. And then the company was spin up to GBIOLOGICS, the company right now we have. So now we are developing it, and we got investment. I think Series A was completed. Our company actually has more experience in consulting because we have vast experience in development of biopharmaceuticals. We provided consulting services as well as producing our own product. And then we have vast network as well as partners. And there's the list of products that we involved for the development, from the proteins to vaccines and chemicals. And currently we have in pipeline, we have 3 products. This anti-cancer drug, as well as RA for escarline product, and then Lucentis biosimilar. Thanks very much.
Okay, thank you for presentation. Okay. Any question or comment? Yeah. So there is one question. Yeah. Warren?
Post-retirement, jdretirementguide.com. No visitors. I'm here as an attorney. in the 60s. Listen to
Well, it's not clear. Maybe Warren, you can send by chatting room, and we can respond to you.
January 27th on NBC. Women Amplified, a Massachusetts Conference for Women podcast hosted by award-winning journalist and author Celeste Sidley. Visit conferencesforwomen.org/podcast.
So, can you show the slide number 11, the KRAS mutant pancreatic cancer?
Yeah.
In that slide, you show that the SCAR 5 mg/kg shows better efficacy than combination with the gemcitabine.
Yeah.
Why is that?
That's very, actually, we expected better with combination, but somehow I think there's interference. I don't know, because the gemcitabine escalane clearly has two different mechanisms. But we are thinking maybe reconfirming these studies to see if there's any interference or the cause of this.
Yeah,
However, it's better than gemcitabine alone.
You should verify this.
Yeah,
This is very
yeah.
unusual.
Yeah.
Okay, thank you.
Thank you.
Okay, now let's move to another next company. That's IMB Dx Inc. So CEO and founder Peter Moon will present this presentation.
My name is Peter Moon in IMB Dx. IMB Dx liquid biopsy company for precision oncology. We are innovating the cancer diagnostics and treatment. We are trying to detect cancer earlier. We capture circulating tumor DNA in blood. We analyze the somatic mutation of ctDNA. We provide information such as which mutation tumor comes from, the effectiveness or resistance of current therapy, and proper medicine for each patient. Through liquid biopsy, we substantialize the personalized precision medicine. We expect the clinical utility of liquid biopsy will increase. The clinical trials of target therapy is over 3,200. The clinical trials of immunotherapy is over 5,000. For target therapy, specific biomarker should be identified. Resistance comes out. In case of immunotherapy, the response rate is less 30%. As the option of target therapy and immunotherapy increase, the accurate diagnosis become more important. As the accurate diagnosis become more important, the market size of liquid biopsy will increase to $100 billion in 2030s. We expect the market will grow up step by step, cancer patient monitoring, prognosis prediction, to early detection. Our founders of IMB Dx began researching of ctDNA and clinical application of liquid biopsy in 2014. Based on the performance of research, founders established IMB Dx in 2018. Our team leaders are experts who can substantialize the liquid biopsy based on NGS technology. Tae Kim was a chief of Seoul National University Cancer Center. Professor Bang has developed the NGS core technology from Chicago and Harvard. Seon Kim is a bioinformatics specialist from Chicago and Berkeley. She participated in TCGA. Tumor biomarker can be DNA, RNA, and cancer cell itself. Most recent approach is sequencing of ctDNA, which can reveal genomic alteration of cancer. The amount of ctDNA reflects tumor burden and size. in early stage, ctDNA is very rare. The most advanced technology is required to detect rare ctDNA in the blood and to analyze signals among noises. Through liquid biopsy, we can profile cancer cell. We can solve tumor heterogeneity issues in tissue biopsy, and we can monitor cancer cell evolution in right time. Through liquid biopsy, we expect that liquid biopsy substitute for tissue biopsy. We've also patented library prep analysis technology, UNIX. We optimize and developed our agent, reagent, and enzyme. We use AI denoising suppressor technology to ensure high accuracy and 100-fold higher denoising suppression. So thus, we can detect and provide a report with as little as 10 nanogram input DNA. We offer a portfolio of 3 products, AlphaLiquid 10, 100, 1000. AlphaLiquid 100 can detect SNP, Indel, CMV, Fusion, and MSI with the 10 milliliter whole blood in 106 cancer-related genes. AlphaLiquid test service takes 10 working days from blood collection to clinical report. AlphaLiquid test can provide the information such as summary of a mutation, longitudinal monitoring, personalized medicine, and information of clinical trials for each patient. While we have many competitors in emerging market, our strengths lie in analytical and detection capabilities with affordable price. We can detect more CMV and fusion with a high degree of accuracy. AlphaLiquid test is validated with many other diagnostic tools. We got same result in ddPCR to detect KRAS mutation in 54 colorectal cancer patients. We have built our own blacklist databases with the analysis of a healthy person. We found the BRCA2 deletion in healthy person. 4 months later, we received a message that healthy person got diagnosis with ovarian cancer. This is one of the examples to show clinical utility of AlphaLiquid test. We could find the clinical utility of AlphaLiquid test in longitudinal monitoring. We can find the increase of somatic mutation burden 3 months before progressive disease. We are developing screening panel for early detection of cancer. AlphaLiquid test is being validated over 3,000 clinical samples in over 10 major hospitals. AlphaLiquid test is adopted in 2 major hospitals for patients. We started to export to Taiwan. Pharmaceutical companies chose AlphaLiquid test for CDX in their own clinical trials. We expect the revenue will increase exponentially as the AlphaLiquid test is adopted in more clinics. The number of tests will increase rapidly. We have two business models. We provide the AlphaRicket test as a service. We sell AlphaRicket package, including kit and analysis software. AlphaRicket test, the most advanced liquid biopsy, can save lives. We invite you to join us in the journey to fight against cancer. Thank you for the attention.
Right, thank you very much. Any question or comment? No? So, well, there are many liquid biopsy tests even in globally or even in Korea. So if you say maybe just one word which distinguishes IBD compared to other companies?
Yeah. Our AlphaRicket test is validated clinically. So that is the main point of differentiation.
Mm-hmm. What's the main reason for the price reduction compared to other competitors?
We optimized and developed our own reagent and enzyme, and we designed our own probes with our strategic partners. So that is the main reason.
Okay, thank you very much.
Thank you very much.
So the next company will be Kanzen Co Ltd, and Park Hyun-sun, BT manager, will present. Okay.
Thank you for inviting us here today. I'm Hyun-sun Park, and I'm working as a researcher in Kanzen. I'd like to take this opportunity to introduce Kanzen. We're trying to make a new paradigm in the cosmetic market with our main innovative technology, which is topical botulinum toxin. Botulinum toxin, also known as Botox, is a neurotoxin secreted from Clostridium botulinum, and its size is about 150 kilodalton. This neurotoxin is widely used both in medical field and cosmetic field, and that is because of its unique mechanism of action. Botulinum toxin works specifically at cholinergic neuron and it cleaves snare protein to block exocytosis of acetylcholine. When the exocytosis of acetylcholine is blocked, it causes muscle paralysis and muscle relaxation. Currently, all the botulinum toxin products on the market are used in the form of injection, so the patients have to just bear the pain of being injected. Besides the pain, the injection of botulinum toxin can be dangerous, especially when it is accidentally injected into the vein. So we question, why does botulinum toxin always have to be in the injection form, which makes it very difficult to treat, especially neckline? And is there any other way to be beautiful in a more natural way? In fact, developing topical botulinum toxin is quite difficult. Because of its big size, the protein does not penetrate under the skin. For transcutaneous delivery, many researchers have tried the physical methods, but we believe that these physical methods can cause skin irritation. So is there any other way to deliver botulinum toxin under the skin in a non-invasive manner? In order to do so, we developed our novel cell-penetrating peptide, which we named as a cargo delivery peptide. Cell-penetrating peptide is a very short peptide which can penetrate cell membrane, and when it is conjugated, it can deliver the cargo into the cell in a non-invasive manner. There was preliminary research that conjugate the light chain of botulinum toxin with the cell-penetrating peptide for transcutaneous delivery. So we decided to develop our novel cell-penetrating peptide, which allows transcutaneous delivery of botulinum toxin. We analyzed pre-research CPP to find optimal characteristic and protein sequence and have developed 13 new CPPs and named as cargo delivery peptide. We tested our novel cargo delivery peptide with known CPPs in silico, and as a result, all CDPs showed greater cell-penetrating ability compared to known CPPs. In order to confirm the cargo delivery of CDPs, we have conjugated CDPs with green fluorescent protein and applied on a mouse skin. As a result, compared to the known CPPs, such as TAT and TD1, our novel CDP showed greater tissue penetration in vivo. After confirming the cargo delivery ability, we constructed botulinum toxin and CDP conjugate and have done in vitro study to confirm its cell penetration ability. In vitro experiment showed that our topical BT protein successfully penetrated human skin cell. Such cell penetration was not shown when BT was conjugated with other CPP such as TD1. Similar to in vitro study, in vivo data, topical BT protein has penetrated human skin while other known CPP products did not. We have done enzymatic tests to see whether our topical BT protein retains the botulinum toxin's ability to cleave snare protein. The results was quite remarkable because not only our topical BT protein retains the ability of botulinum toxin, but also it showed about twofold greater enzymatic activity compared to the normal light chain of botulinum toxin. Also, we confirmed the safety of our topical BT protein by doing WST1 assay with human skin fibroblast, and the results confirmed that topical BT protein did not show any toxicity to skin cell even when it is applied in the high concentration. With our topical BT protein, we're making a new innovative anti-wrinkle cosmetic product. We have done small-scale clinical tests to see anti-wrinkle effect of topical BT protein. In order to do so, a participant applied the commercially available anti-wrinkle products on the left crow's feet, and on the right side, participant applied our topical BT protein. And we measured wrinkle depth and the total number of wrinkle by using optical 3D measuring system. When participant applied both control and topical BT for 2 weeks, the control showed no difference in wrinkle depth compared to its week 0 data, but our topical BT protein caused 136 micrometer reduction in wrinkle depth. In terms of the total number of wrinkle, at week 0, the total number of wrinkle was 347, but when the topical BT was applied for 2 weeks, the number has decreased to 54. The anti-wrinkle effect of topical BT protein was showing in the first week of applying, and when the topical BT was applied for 4 weeks, it caused a maximum 155 micrometer reduction in crow's feet's wrinkle depth. And this result was not shown when the commercially available anti-wrinkle products was applied on the same person. Next, we increased our topical BT content up to 20 ppm and applied for a short period of time to test its effectiveness. When 20 ppm of topical BT was applied for 3 days, it caused 238 micrometer reduction in crow's feet's wrinkle depth, while control showed no difference. This was also effective and evident on under eye wrinkle, which caused 246 micrometer reduction in wrinkle depth. With this result, we applied for Korean patent, and patent has been approved and registered, and also we applied for PCT. Our topical BT protein has been approved and registered in International Cosmetic Ingredient Dictionary, and our work has been recognized and selected as a government-funded R&D project. Kanzen CDP technology has huge potential as a platform technology because it can be used to deliver many small molecular drug, protein drug, and nucleic acid drug. In fact, it can be used in many research areas such as cancer therapy, brain drug delivery, and gene therapy. As of now, the global botulinum toxin market is shared by few key companies, and this 8 million worth of market is becoming a red ocean. But with topical botulinum toxin and CDP technology, Kanzen will become a game changer in the global market. Our company is run by top-tier human resources, especially Dr. Choi, our CEO and CTO. He received his PhD in biomedical engineering and biochemistry from University of Massachusetts, and he worked at U.S. Army Native Soldier Research, Development, and Engineering Center to study botulinum neurotoxin peptide inhibitor. Also, we are collaborating with many plastic surgery specialists for clinical research. Besides, many highly qualified and experienced researchers are working with us. Our research center and office are located in Seoul National University, and we have a certified BL2 lab where we handle our botulinum neurotoxin experiments. We have a huge R&D network within Seoul National University with Proteome Tech and Chungbuk Technopark. Like our company's mission statement, we will continuously strive to give you great pleasure and hope through the implementation of innovative biotechnology. Thank you for listening.
Thank you for presentation. Any question or comment? No. So, is there any other company which is developing topical botulinum toxin globally?
Globally, according to our research, globally, we believe that Revance Therapeutics, they tried making topical botulinum toxin for therapeutic use, but they went up to, they passed phase 2 clinical tests, but they failed a phase 3 clinical test. And according to our research, we believe that they tried to conjugate botulinum toxin, and they also use CPP. And they conjugated with just ionic interaction. So because of that, we're trying the covalent bonding with botulinum toxin and CPP, and we're expecting more effect with that conjugation technology.
Well, so I saw that you have a dramatic effect between the control group and your topical botulinum toxin group. Can you design the experiment which left side you can use injection, and the right side you can use topical, and seeking the comparison data between the injection and the topical application?
We have not tried that yet, but
That's
we believe that we
maybe
can.
meaningful data for the same person, different administration route. If you show some difference or superiority compared to IV, that would be much better, strong maybe data.
Yeah, sure. We absolutely need that data, but before that, we will need to determine the content, which content would be comparable with the injection type.
Sure. Okay, thank you very much.
Thank you.
All right, the next company will be Oncorcross, and so Daniel Kim, CFO, will present his company.
Thank you. My name is Daniel Kim, CFO of Oncorcross. So Oncorcross is an AI platform-based biotech that helps pharmaceutical companies to develop and accelerate drug development. Our mission is to increase the number of drugs in the market with lower costs in a shorter time. Our core competitiveness is that we can enter clinical trials quickly, unrivaled database quality that can improve the success rate of clinical trials, and we know the healthcare market better than our competitors. Everything starts from this graph. The graph shows the number of FDA-approved drugs per $1 billion spent. In 1950, nearly 100 drugs were approved and came to market. But by today, it is less than 1. In fact, according to the research, pharmaceutical companies on average take almost 10 years and spend $2.6 billion to have one drug approved. As stated before, our mission is to increase the number with less money in a shorter time. And let me give you why we think this is possible and how. Most of AI biotechs are in the early stages of drug discovery. This area requires heavy R&D spending, and frequent failures are inevitable, whereas we focus on the later stages of development. We take the drug compounds that are beyond preclinical stages, and using our AI platform, Raptor AI, we found new indication. Once we identify the new indication, we can skip the discovery phase and start clinical trials. This way we can save up to 8 years in time and 95% of the cost that is typically incurred to reach clinical studies from the early stage drug discovery. Then how do we identify a new indication? and this is where our database comes in. Diseases are caused by complex interactions between many genes. However, majority of the pharmaceutical companies focus on a single gene target. And we think the focus on a single target is insufficient to treat diseases properly. Therefore, we take a holistic approach by analyzing the gene expression pattern to reverse the unbalanced gene back to the normal gene. In our proprietary database, we have over 100,000 patients' transcriptomics data, over 25,000 drug signature data, more than 410 different types of disease, and 40 types of cancers with various phenotype information. Drugs and disease data collected are standardized and normalized to yield RNA expression data. And then our AI platform, Raptor AI, analyzes data to result in new drug indication, additional disease indication, and combination therapy. Raptor stands for RNA Expression-Based Anti-Symmetrical Pairing Tool for On-Demand Response. We apply Raptor AI in 6 different business areas. In indication expansion, we screen new or additional indication for a drug compound in preclinical or early stages of phase 1. In cancer, we screen to find the optimal indication or biomarker for anti-cancer drugs. In rescue drug, we screen to find new indication for compounds that have failed during phase 2 or 3 clinical trials. In IMD, which stands for incrementally modified drug, we find the best combination of 2 drugs to improve the efficacy synergistically. And in orphan drug, we screen marketed drugs to find rare disease indications. And in infectious disease, we screen marketed drugs timely to find optimal drug combination during pandemic situation like the COVID-19. And let me give you one example of our value in real-life application. Sarcopenia was coded as proper disease by WHO in 2016, but there are no approved drugs in the market. Using Raptor AI, we found OC501 and OC504 to be a good drug combination. After we validated it in vitro and in animal model, we licensed out to Korea Pharma. Now, this records the first licensing out deal of an AI-initiated drug candidate from an AI biotech to a traditional pharmaceutical company. Korea Pharma is to conduct clinical trials soon, and this was done without spending millions of dollars in many years. We provide our value in other areas of business. In indication expansion, GL Pharma was developing a drug candidate with stroke as indication. We screened and analyzed that this can also be used as a treatment for a specific liver disease. We helped GL Pharma to add one more pipeline while saving both time and cost. In cancer, ST Pharm was planning a clinical trials of an anti-cancer compound with 5 different types of cancers. However, after screening on our AI platform, ST Pharm redesigned the study by replacing one specific type of cancer indication and began clinical trials in the US last year. This way, we helped them to increase the success rate of their new anti-cancer compound. The IMD example was given in the previous slide. In rescue drug, we received 5 compounds from Boehringer Ingelheim that failed during clinical trials. We found 1 compound showing a meaningful indication, and we are currently validating in animal model. At the same time, we're also discussing with the BI on the next step forward. In orphan drug, we can screen and repurpose existing drugs with known pharmacokinetics and pharmacodynamics to find a solution for rare disease with unmet medical needs. This way, we're collaborating with the Severance Hospital in Korea on a rare infantile epilepsy and planning clinical trials next year. We're also collaborating with the Columbia University on other rare diseases. Finally, in infectious disease, we're collaborating with the Joseon University Hospital to find a combo therapy for current COVID-19. As you can see, Oncorcross' AI platform and unique business focus allows us to identify the target indication and drug with high accuracy. This way, we can advance into clinical trials rapidly at a low cost, whereas most of the other AI biotechs are in the early stages of drug discovery with high R&D cost. At the end of the day, if the compound does not pass the clinical trials, it cannot become a proper drug. Currently, we have 15 pipelines, 9 in-house and 6 partnered, and among these, 5 are to begin clinical trials within the next 12 months. This makes an attractive partner for the big pharmas. Currently, we are collaborating with Cancer Research UK, Columbia University Medical Center, and Boehringer Ingelheim, and many more. With these partnerships, we focus on 3 markets: oncology, no drug, and failed drug market. Global oncology market is estimated to be 150 billion, and it's growing. For orphan drug market, it is estimated that there are 7,000 rare diseases globally, and less than 6% have approved treatment. And finally, on average, 86% of the drugs in clinical trials fail, and billions of dollars go wasted every year. We know these markets better than anyone else, because Oncorcross management largely consists of healthcare professionals. We have 3 medical doctors, a pharmaceutical chemist, and 2 pharmacists, led by Dr. Irang Kim, the CEO and a medical oncologist. This makes us more a healthcare market-driven company, since we know where the unmet medical needs are versus the other AI biotech, which most of the founders have IT background. Now let me repeat this again. Accelerate drug development with Oncorcross. I hope this one-liner makes sense to you. Thank you very much for your time.
Okay, thank you for a very nice presentation. I don't see any question or comment from the audience. So, Oncorcross is different from other AI-driven development company.
That's right.
So you are in the late stage.
Yes.
Any other company globally aiming for that target?
I believe there are, I'm not sure about the number, but 80% of the AI biotechs are in the early discovery phase. Remaining ones they're scattered in development phase focusing companies. I believe there are a couple of ones in the US as well. Helix would be one example. And Recursion and Benevolent AI, they actually take the whole areas of drug discovery to the development phase as well. And in Korea, we are the leading drug development AI biotech. I believe there are 1 or 2 more that are trying to catch up with us, but in terms of database quality, database numbers, I think we're far ahead.
So you mentioned that you have a collaboration with the Joseon University Hospital for COVID-19 project.
Yes.
I saw one Korean AI company did that kind of screening job earlier. Or you have any kind of good data so far?
I believe the company was SynthegaBio, the COSAC listed company. Their database levels are at a genomics level, which is at a DNA level, whereas we're at transcriptomics, so we're at the RNA level. Getting a RNA data sample actually is quite challenging. So our CEO been contacting various big hospitals in Korea, but was unable to get the actual blood samples for the COVID-19 patients. So that's why we are collaborating with the Joseon. In terms of SynthegaBio, I'm not sure how they actually approached and analyzed the data, but we're actually slightly different.
Okay, thank you very much.
Thank you.
So next presenting company will be Onegene Biotech, and Dr. Sungjin Park, CEO, will present. Okay.
Thanks for giving us the chance to talk about our technology and assets. We are developing a microenvironment targeting drugs for the complex diseases such as organ fibrosis and cancer. We believe the synergistic targeting of these older players in the fibrotic microenvironment will make a big difference in treating patients suffered from those diseases. To do that, we developed the Unistack technology, which allows developing the protein drug harboring more than 3 targets. And using our NASH assets, we have been successfully showing our POC for such a transformative drug concept and the Unistack platform. First, I will talk about the NASH, then our platform Unistack, and our pipelines. Many adults in the United States are affected by NAFLD and NASH. NASH is the fastest growing cause of a hepatocellular carcinoma and the most rapidly increasing indication for liver transplantation in the USA. Presently, there are no treatment approved, especially due to the high difficulty in fibrosis resolution. NASH differs from simple steatosis or fatty liver by the presence of the hepatocyte death, inflammation, and various degrees of fibrosis. There are key targets at each stage. At first, lipotoxicity and apoptosis, then immune cell-mediated tissue inflammation, and then the activation of a hepatic stellate cell that differentiate into myofibroblast. This means that at the fibrosis stage, all the problems are interlinked and coexist in the microenvironment of the fibrotic liver. This is the simplified view of the fibrotic liver microenvironment. Traditionally, NASH drugs were developed to target only the bicide chunk from this whole picture. During 18 to 19, we witnessed many failures of such efforts. In 20, the year of a bright future, the promising drugs that both resolve NASH and fibrosis were finally introduced. One of them are FGF21 and 19 derivatives. The key distinction was that these drugs target multiple aspects in the fibrotic microenvironment, both directly and indirectly. We wanted to further enhance such a holistic effect of FGF21 by installing more ligands, targeting macrophage and stellate cell. In a way, we aim the best-in-field drug that can resolve both NASH and fibrosis better and faster than any other drugs. But we needed a technology allowing connecting these 3 or more clinical well-validated targets into a single molecule. And the resulting molecule must secure high developability, especially stability-wise, and degree of freedom for connected ligands, and the commercial feasibility, of course. Although we talked to many CROs and we did our own research, we failed to find any to satisfy our such criteria. So indeed, while there are 650 monospecific and 56 bispecific drugs on the clinical trial and market, but only 5 for trispecific drugs in the clinical stage. For elucidation, we conducted our research on the trispecific platform people use. This is the prevailing trispecific format. It is difficult to make. Actually, it's a tough challenge for a single cell to be optimized for simultaneously expression of the 3 ligand and the following heterodimerization. The resulting structure gives a highly crowded space above the hinge region, therefore often lowering efficacy of the second ligands, sometimes completely ablating it. In many cases, these complex structures give lower stability, which is the most common developability issue for the multi-target drug development. It was obvious to us that a dedicated platform for 3 or more ligands must be introduced. So we tried many ideas to meet our criteria, and after years of work, we realized that we had to say goodbye to traditional FC-based, single-cell-based, or gene-level fusion-based approaches, which have been our comfort zone for decades. Unistack is a post-translational fusion, meaning that we produce 2 pieces separately and then fuse them later. This is an enzymatic conjugation that features near 100% conjugation yield with an unprecedented atom economy. And separated expression allows higher flexibility in designing the 3rd ligand structure. It also allows true plug-and-play practice when we try to find the best ligand combination. This is really convenient, and it saves so much time and the money. This is the actual before and after image of the conjugation. Here's the albumin part before and after, and there's no albumin part remaining, unreacted. So we observed these results for all the pipeline we tried so far over and over, which means it's pretty general. Unistack enzymatic conjugation yields the rigid scaffold that separates the ligand by 4 nanometer. It is 10 times more spacious than corresponding hinge region of the Fc-based platform. Furthermore, the resulting conjugation produces a branched three-way structure, not a head-to-tail structure as in a traditional gene-level fusion. So why is it important? Three-way conjugation secures free ends for each ligand that is added. Therefore, it secures the degree of freedom for each ligand movement. And it's important to minimize the thermodynamic penalty that happens when ligands are connected to other ligands or carrier protein. Most importantly, all the unistack pipelines show some stability with a TMT aggregation over 70 Celsius, which is very satisfying number for molecules with this level of complexity. Finally, using unistack, we developed the two NASH assets. We installed the ligands targeting three major players in the fibrotic microenvironment of the NASH liver, which is hepatocyte, macrophage, and hepatic stellate cell. All the targets were clinically well validated. The MCD mouse model is to determine how effective the drug is for the fatty liver and the steatohepatitis. All the mouse, 100% of mouse with the OGB-21502 resulted in non-NASH or steatohepatitis free. The difference in NASH score from the control was 3.4 versus 1.1. CCL4 model is one of the harshly induced fibrosis model. OGB-21502 prevented the cirrhosis progression from 80% of the mouse. Fibrosis score difference between the control and OGB-21502 was 4.0 versus 2.4. These two data suggest that OGB-21502 is potentially the best in field FGF21 asset in resolving both NASH and fibrosis. Interestingly, other organ fibroses have similar microenvironment and players. Using unistack, we'll make a different combination of the clinically validated targets to produce 4 pipelines. These pipelines will be tested in parallel with the corresponding disease models. We will know the efficacy profile that will allow us to select the best candidate for each organ fibrosis. Large body of evidence suggests that the tumors create an immunosuppressive microenvironment that inhibit the immune system's natural ability to recognize and destroy tumor cells. Because the immunotherapy responses only occur in a relatively small fraction of the patients, tumor microenvironment targeting is gaining huge attention these days. We totally agree with this direction, and we will have our own POC, immuno-onco POC, by this year. We have a short-term goal of introducing 7 tri-specific drug pipelines by 2024, and one among them will go into the clinical trial. Currently, there are only 5 tri-specific drugs in the clinical stage. After 2024, when our first line enters the clinical trial, we will become the most highlighted company in the class of tri-specific drugs. Conclusion, we develop a microenvironment targeting drugs for complex diseases such as organ fibrosis and cancer. The unistack is a dedicated multi-targeting platform with a commercial feasibility and superior developability. OGB-21501 and 2 are the first unistack drugs that simultaneously target hepatocyte, Kupffer cell, and stellate cell in NASH microenvironment. Microenvironment targeting immuno-onco pipelines are our next expansion. The complex and chronic diseases like fibrosis, cancer, and even CNS is hard to cure, and they bring so much suffering to all of us, our family and friends. Onegene is the first company proposing synergistical targeting of as many targets as possible in the disease microenvironment can provide a new opportunity to help patients. This is a simple idea, but it's never been tested. In this phase, we develop a dedicated platform to develop protein drugs targeting 3 or more. I hope we can find a good partner to join our endeavor to solve this big problem together. Thank you, and I will be very happy to have any questions.
Thank you for presentation. I don't see any question or comments again. So you mentioned that there are 5 tri-specific companies in globally. So among this one, what will be Ongene's most priority difference?
Yes, the most important difference will be the speed.
Speed.
Yes, because our platform first applies this post-translational fusion, which means that like we can make a library of one side and the other, and then combinatorial testing, which is most important for multi-targeting drug optimization, will be uncomparably fast. So 3, 4 is going higher. Our platform gives relative higher advantages.
Okay. Thank you very much.
Thank you very much.
없어. All right. Thank you very much, and good luck. Okay, the last company, the last but not the least, is WellsCare, and Junie So, Global Sales Director, will give a presentation. Okay.
Thank you very much, everybody, for inviting me online here. It is an honor for me to be pitching at this Global IR presentation all the way from the Eastern Standard Time. So thank you very much. It's the last company that I'd like to introduce you, and that is WellsCare. And let me just upload my slides. Thank you. Now, WellsCare brings you your everyday care, but now a bit smarter. The problem that we try to solve actually comes from a rise in technology. It's an unusual problem because usually we think a rise in technology will bring more benefits. For example, the 3.8 billion users we expect to be holding onto smartphones by this year. This rise in technology actually brings in painful aspects such as DQT or texting thumb, which apparently 50% of users under 19 already are subjected under. So this 3.8 billion users, including myself of smartphones, am subjected to texting thumb pain. And the 1.5 billion devices in addition we expect by next year will be subjected to VDTS. Now, why is this becoming a significant problem? It's because this bad posture lifestyle in front of our devices tend to bring types of pains that we deem inaccessible for treatments, meaning it's not really worthwhile to go to the doctor for the shoulders pain and the neck pains that I have. And that is why now we see a significant increase of people with carpal tunnel syndrome, for example, people with tennis elbow or golfer's elbow, and even people with plantar fasciitis, just to name a few of these disabilities. Now, we looked at the problem, and the problem is pain. And pain usually has 3 main components on how we try to treat it. The first one is we go to the major hospitals and look at the pain specialists and see what they have to offer. It's effective, but usually it's expensive and requires heavy commitment in time. Now, that is why some people go to clinics instead, which is more accessible, such as physiotherapy and massage, but it also can be costly, and the efficacy of pain relief can be questionable. So the rest actually try to take care of it at home, where they give themselves self-therapy, which is perhaps the most low-cost point, but it can actually do more harm, and it can be really ineffective in terms of the pain. So what we wanted to do from WellsCare is to bring in the 3 aspects of this and only bring in the good, something that it can be effective, accessible, and low cost. And that is where we bring you the solution to our problem, and that is IASO. WellsCare brings you IASO, which is a very simple to use, compact, only 48 millimeters in diameters and 12 millimeters in thickness, a wearable pain therapy device. And it uses a market-proven, effective pain therapy technology now accessible at home for all modern-day people to utilize. Now, the therapy solution behind it has actually already been in use in the hospitals and the clinics, but they've been bound by large, bulky machines that has a very high capital expenditures for the hospitals and the clinics. And even for the patients, you have to have numerous visitation and also pay for a lot of money for this type of treatments if they don't have insurance. What we did is we brought the same medical grade technology, now accessible as the world's first hands-free cold laser inflammatory pain therapy device. It's a mouthful, but it is a device that is on par with hospital grade technology and that is capable of taking care of the pain on your shoulders and your neck and any other type of inflammatory pain that I mentioned before. EASO as a product, as a product compared to the other massagers and pain therapy devices compares best when you look at pain reduction, where we have clinical trials and scientific papers to show its high efficacy in pain relief. And of course, we are proud to say we are the first truly wearable, truly hands-free device to be utilizing this technology and no side effects. Now, let me just briefly explain to you how our device works. EASO will sit on top of your skin at a pain-affected area, and the two lasers will penetrate the skins. Now, the two specific wavelengths of laser we chose have been shown in science and many papers to have the most effects in vasodilation, dilating your blood vessels. Our device also heats up to 40 degrees Celsius, which is slightly above body temperature. So more heat with larger blood vessels mean higher blood circulation. And that is a simple step behind EASO. With more fresh blood in a pain-affected area, the cells can now have more access to ATP. Higher ATP generation means more cell regrowth and regeneration, and therefore decreasing the pain factor. It's not a new science tech, but now it is accessible for everyone through our device EASO. It is scientifically proven for over 100 years, but we didn't want it to stop there. We wanted to make sure our users and our investors feel confident about our device. So we've published we conducted two clinical trials, one to be published this year, where we found three main key points. The first one is that the pain reduction capability is 88.8%, meaning out of 10 people with pain, 9 people will feel the pain reduction. And we've also found that only with 8 minutes of use, they will feel a 30% of pain reduction. And the final fact that we're really excited about is that our device is not only effective for people with acute joint pains, but even with chronic joint pains. We are, of course, protected to be the only and best WellsCare in the world with 17 patents and more upcoming. And we have qualified trust from the FDA Class 1 registration, FCC, ICC, and CE, so that our device is ready to puncture globally. Our device, of course, comes with a team of experts who have more than 17 years of cold laser expertise, devices that you have usually been seeing on clinics and hospitals, but now we bring you, with the medical professionals, a device that you can bring at home to your door. So that was our first step, and we found that with tremendous success in all of our online and offline sales, as why we are available on Amazon, on Brookstone, and Newegg, just to name a few. And through this process, we have successful customer acquisition, and we have successfully built a brand around pain management. And that was all done through this hardship of 2019 and 2020. And in our first step, we found that directly to our customers, bringing this therapy form accessible to the comfort of your home was very successful. But we think we can do more, and that is where our B2B step lies in. Now we can decrease the client to practitioner ratio, allowing the clinics and the small physiotherapists to increase their profit margins. And we can bring the same therapy solution. So for our next step, we want to introduce you EASO Pro. EASO Pro will target the medical clinical level providers. And right now we're looking at the 250,000 health practitioners in the United States just to see how they'll be able to utilize our device into their patients. So as we were doing a lot of our marketing efforts in our B2C sales, we've already made relationships with health practitioners in clinical settings, and they actually are our first target market on onboarding the first 20 B2B medical experts, and we expect that by Q2 of this year. So we were expected to enter a market that was supposed to reach $46.6 billion, and this is for the world pain management and drug and device market. Now, I know that COVID has changed its numbers, but the efficacy on us penetrating this market is anything even higher, I think, because of its ability to be utilized at home, and more of us are looking for alternative solutions at home without going to the major institutions. Now, this is a very large chart with numbers that I'm willing to come back to during our Q&A, looking at our projected sales in Korea and, of course, in North America, our main market. But this projection cost is what we have. ESO from WellsCare has already been funded in our pre-series with our wonderful supporters who has valued us up to a post-valuation of 5 million, raising up to 750K. And now we're looking for a Series A partner for a post-valuation of 12. And that's what I have for you today. My name is Junie Su from WellsCare. And for a life without pain, please consider finding our product at wellscare.net. Thank you very much.
Thank you for your presentation. Any question or comment? I don't see any. So you are currently selling ESO on the market?
Yes, sir.
What's your annual sales now?
So we started selling ESO in the North American market as of Q4 of 2019. So the only real year we've been able to sell ESO on the North American market was last year. And we're proud to say we're just shy of 1 million.
Okay. So you mentioned that ESO gives heat regeneration and up to 42 degrees. So how long it lasts?
How long does the
high temperature?
I see. So our device throughout the 30-minute session will be heated up to 40 degrees Celsius. So it will always be heated up. Is that the question?
So for 30 minutes. So after that,
yes, sir.
It drops to the normal.
Correct.
Okay. All right. Good luck for your ESO Pro. Okay.
Thank you very much.
Well, so this concludes the 2021 Global IR pitching at JPM. I hope this pitching is your opportunity to advance for not only all 8 presenting companies, but also for all online other participants. Another in-depth panel discussion for Korean market and 8 global biotech startups will be ready for tomorrow's session. So please join us tomorrow at the same time. Okay? Thank you very much.